here, and then we'll switch spots.
They can hear us.
Oh, I forgot. Okay, we're going to get started, everybody. Can you all hear us? Yes. Okay. So, hello, welcome to Grouper Moon 2016-17. I'm Todd. You guys, I've been emailing with a lot of your teachers, and we're excited to have our first live stream today. And I'm going to quickly tell you what we're going to be doing. I'm going to introduce you to the two primary scientists on the project. They're going to give you an update about what's been going on. I believe that we're going to have a special visitor in a little bit to talk. We'll see if that happens. And then I have another scientist who's working with hydrophones, she's gonna explain the project she's working on. And then if we have time, we have another special guest who has been working on this project since she was just about born. So that's what's gonna go on for right now. So first, I'm gonna introduce you to this is Bryce Simmons, and he is a reef scientist working on the project, and then Christy Pattengill, and I'm going to hand the mic over to her.
Okay. All right. Hi, I'm Christy, as Todd said. As Todd said, we've been working on this project for about 15 years. Yeah, 2001. Unmute.
They can hear you. 15 years.
I work for the Reef Environmental Education Foundation. That's a nonprofit. We're based in Key Largo, but we do work for marine conservation all around the world. And this is one of our key projects that we do every year. We're really excited to be able to share it with you today. We arrived here a week ago Tuesday, and we have been busy getting all set up for, we have a lot of different experiments that you're going to learn about today and over the next couple of days to try and study these large spawning aggregation off the west end of Little Cayman Island where we are now. So.
Yeah, great. So you guys, I think, have talked at least a little bit in the classrooms about the project that we're doing here. But just to remind you, the main thing we're doing here is we're studying a spawning aggregation of a species called Nassau grouper. You guys know what that looks like, right? Nassau grouper are a really amazing species. They're one of the top predators on Caribbean coral reefs. So they're really important in an ecological context, and they're also, at least in a historical context, they're quite important socially. People like to catch them, people like to eat them. And they also have this habit of getting together in large aggregations in order to reproduce for a short window of time. It's really predictable in time and space. And in the Cayman Islands, that happens right now, either in January or February, starting around the full moon to about 8 or 9 days after the full moon. All of the fish around the islands go to a single spot or maybe a couple of different locations. It's really, really small. They all crowd together, kind of like you guys are probably crowding together right now in your classroom. And that's where reproduction for the population happens. And that's it for the entire year. That's the only place they reproduce. But the problem with that is potentially that if you find it as a fisherman, it's very, very easy to remove most of those fish very quickly. And that's what's happened throughout most of the Caribbean, except in a few places. And here in the Cayman Islands, we're lucky in that we have the largest known spawning aggregation remaining for the species out here in Little Cayman, and some other large aggregations that are still functioning, including one that we just got a report back from researchers over on Cayman Brac, that they literally just got up from a dive about 10 minutes ago, and they reported that they saw maybe 1,000 fish or more over there in really nice conditions on the east end of Brac, just beyond the bluff. So really, really exciting stuff. And that's part of the really cool thing about this project is we've got lots of different things going on, not just over here on Little Cayman, but on all three of the islands. And exciting stuff is happening, not just here, but everywhere throughout the Great Cayman Islands when it comes to spawning aggregation research. So really excited.
So, So the last couple of days, we've seen spawning here on Little Cayman. We had spawning starting on Monday night, and the really big event of spawning was last night, which was Valentine's Day, which is a pretty exciting coincidence. We expect there to be spawning tonight and tomorrow night as well. So we've been doing a lot of monitoring out there, documenting how many fish there are, but also capturing egg collections, doing egg collections to look at genetics and connectivity between different places.
Why would you need to know about the genetics?
Right?
How does that help you?
Yeah, so I'm gonna go ahead and hold my microphone closer to my mouth. Hopefully this will help clear things up a little bit. Somebody type and let me know whether or not this is more helpful or not while I continue to talk. So Todd had asked. All right.
Hi.
Hi. We've got a classroom meeting with us. Hi.
You guys stop.
Okay, go ahead. Is there a question?
Yes, it's supposed to be on mute.
Yeah, you guys are. So you were on mute before. And,
You guys, I really need you quiet.
Mute now.
Okay.
Don't sit on the ball if you can't.
Now, if you guys are, if you've got a question, go ahead and ask it. Otherwise, we'd ask you to mute the microphone. And
Yeah, Martha, go ahead and mute for right now. We're going to just give an overview of what's been going on. Okay? Hi. All right, I'm going to mute you until you figure that out. And then, Bryce, you're going to go ahead and continue talking about why it's important that we're studying the genetics.
sure, we can still hear them, Todd, but I'll go ahead and it sounds, oh, there they go. They're not muted. Okay. Okay. So two things. First, I'll say I know that at least a couple of your classrooms last year joined us, and last year when you joined us, there weren't that many fish. Well, this year, let me tell you, it's a completely different story. The last couple of days, we've had thousands and thousands of fish out on the west end of Little Cayman, many more than we've ever seen before. So that's really exciting. What's also really, really cool is that a lot of the fish that we're seeing out there are these babies. They're about this big. They've just become reproductively active. They're probably only about 5 years old, and this is their first time going to the spawning aggregation. So these are the teenagers in the population that are just now starting to show up. So it's really exciting. Todd had asked earlier why we care about genetics. And we use studies on the genetics of these animals to look at population connectivity. And why does that matter to us? Well, we need to know, or we would like to know, to what extent populations here in the Cayman Islands are related to other populations throughout the Caribbean, like, say, for instance, Cuba, because that helps us to get an understanding not only of where fish are coming from when they show up at these aggregation sites and show up as juveniles on our islands, but probably more importantly is what other areas in the Caribbean basin are the spawning aggregations that the Caymanians have that are actually seeding other locations. Because you guys are lucky here in the Caymans that you have such a large spawning aggregation. You also have the potential to be the epicenter, the place where the population starts to recover by spreading throughout the Caribbean basin. And we can ask and answer all of those types of questions by looking at genetics to determine how related populations of the species there are between regions.
Awesome. We have a bunch of classrooms that are joining us that have been part of the program for a few years, and we've attempted to do some live dives from the SPAG, but the last 2 years we haven't been super successful at finding the fish. What's different about this year? Why do you think we weren't able to find them last year, and this year it seems that they're here? Can you explain that a little bit?
Yeah, so I said that they're very predictable in time and in space, right? We know when their spawning aggregation is going to happen, mostly. It turns out that if the full moon happens in January and or February, and it's halfway between months, so maybe it only happens in the middle of January and again in the middle of February. Well, we don't know if the fish are going to show up and spawn in those months. It could be January, it could be February, and so we have to guess because we can't be down there for both months. Last year we guessed wrong, and we showed up, and so a lot of the fish were there, but just not all of them, and they didn't end up spawning on that month. This year we guessed February, and because of the way the moon fell, we were more confident in our guess, and it turns out we got it right. So that's really good news. So point is that while they are very predictable, even us as scientists, having spent almost 2 decades studying this population, getting it exactly right is still at least a little bit an art as opposed to all science.
Awesome. Well, so we have a special guest that I'm going to introduce to you guys. Guy Harvey is going to come on and talk about how he's been involved in the project. So let me switch spots with him. You're going to talk a little bit about your part in the project, and then I'll prompt you.
So I can introduce a little bit first to start. So a lot of you guys, you probably know who Guy Harvey is. He's no stranger to local schools in the Caymans, throughout the Caymans. Guy has also been part of this project for, gosh, going on
7 years.
7 years,
Yeah.
no kidding. Guy's really been an integral part of the project. He not only comes out and helps us with the science week. Today we had him out collecting data for us, doing counts of tagged to untagged fish. That's part of one of the things that we do out there. So somewhat mundane, but science needs to be done, and we need competent divers to help us with that. And guess what? Guy's one of those. But Guy also has a group of people that help with collecting images and pictures and videos and all kinds of cool new and emerging film technologies that helps us to document what's going on out here, not only just to document the amazing spectacle that it is, but also to put a story into place that helps to tell what we're doing and give outreach associated with the project.
Well, that's true, Bryce, and it is a fantastic story to tell. It's a story of success, of the collaboration between many different entities over a long time to get this all to work and to tell that story. So the storytelling part is really important. We're glad to help, on a piecemeal support basis, but it's really important that this get out to the rest of the world, and that's also why it was so important to have Sir Richard Branson come down here and actually see it for himself, because the other part of what you're doing, which is so great, is to export all the experiences, all the knowledge to other Caribbean countries and to show them that it is doable, it does work, and these populations can come back.
It's great.
Yeah. They don't know that Richard Branson was here. Can you talk a little bit about how that happened?
So Richard Branson is a good friend. We have collaborated on several other projects in the past, on sailfish, on whale sharks, on mako shark research work. He's a keen supporter of all that's going on in the Caribbean and has really been the glue of what we now call the Caribbean Challenge Initiative. But there's nothing like taking people to a place to show them the effect of good management and good collaboration, good enforcement. So I invited him last year, and we planned this from 10 months out, and he shows up of the week and really experienced the very best of this spawning behavior.
So you've been here for about 7 years. You've seen a lot of this. What do you think that's new and interesting about this year?
Well, the key thing was that the first reason we did the first group in this documentary was to get the kids in Cayman familiar with what the SPAG was all about, but also to use it as a tool to educate them about the fact that SPAGs need protection. And that year, 2011, the restriction on the SPAGs was going to get lifted. And so we took the documentary that George made throughout all the schools. We got the kids to write letters to the minister then to say, you've got to extend the protection to the SPAGs. Not only are the group as important as medium reef predators, but also they can deal with the potential threat from lionfish.
Some of these students actually wrote letters.
Yeah. Great. Well done, you guys. It worked. The next thing was the next year was a banner year, because we saw the juvenile groupers up in the back reef here, these 3 to 4 inch long groupers, and nobody here had ever remembered seeing the little baby groupers around the place. So it was with great excitement this year, after 5 or 6 years, we were expecting to see them show up as the new adults on the rock, joining the broodstock on the SPAG. And guess what? We've seen more than 1,000 new fish, Bryce, I think, come and join the broodstock, which is, it's so important. This is the capital with which we're going to see, you know, fish for the next 2 or 3 generations of fish just absolutely start to really increase numbers. I hope it really goes exponential from now on because of the volume of eggs that we're now seeing out there.
Yeah, and that's, we talked about that a little bit earlier. It's those teenagers that we talked about showing up, and it's really amazing to see that pulse of young fish showing up 5 years ago on our back reefs and up in the mangroves and in the seagrass beds.
And we caught a few. We got DNA from them.
Yeah.
We did a film
And
on them.
like that big, right? Yeah.
They were great.
And watching that group of young fish slowly turn into adults, it's a pretty amazing thing for sure.
So the aims and goals of this documentary, of course, 6 years later, is to tell the story of what has transpired, how your science has improved, the ability to collect data, and of course, the long-term nature of this data is becoming increasingly important because you can detect fluctuations in populations. You're doing new stuff in detecting eggs offshore to track where they go in the drift, because these eggs have to come back to land at some point, I mean to the reef at some point. And that's all great. The involvement of the Department of Environment, of course, has been consistent for all this time, and so we're telling the story of collaboration, and it's just a great story to tell, and you know it's really exciting to see the Bahamians adopt it, to see the people from BVI adopt it, and who knows who else in the Caribbean is going to say, hey, the Caymanians did a great job, let's follow their lead.
So, do you have a moment to stay with us?
Yeah.
I have.
I could also put Jessica on if you want.
Well, we have kind of a filled schedule. Just one second. I want, because you're going to be able to answer the stuff that. Right. Okay. Hey, everybody. Hi. So I have a video that the researchers took last night of the spawning, and I'm going to pull that up for you guys to see. And then when we come back, Bryce and Guy Harvey will still be here, and I'm going to give you guys a chance to ask them some questions. So while you're watching the spawning video that I'm going to pull up, I want you to be thinking of some questions that you would like to ask. I'm just scooching forward to a couple of highlights for you guys. That's a lot of fish. So then when we come back, we can talk a little bit about that behavior. And here's another one.
That's a good one.
You can see them collecting the egg samples. Okay.
There's a good one. Fireworks.
That is fireworks. It's beautiful. Okay. They couldn't see it before, so we're going to go through this again. Okay, so we're going to go to another highlight. Sorry you couldn't see it before, but we got that fixed. And sorry about the wind. The last one's the best. We're going to come right up on Steve.
Just missed that one. There you go. That was it.
That was it.
George, you need that one.
Okay, so. All right, here we are back. So I'm going to hand this microphone back over to Bryce, and you guys are going to be able to ask some questions.
Can they unmute themselves, or are we controlling the mute?
They can, and so let them know they can unmute themselves, or they can type it into the chat.
Okay, hi everybody. Should we take them one at a time? Todd, tell them who's
Yeah.
on.
So if you have a question, borrow your mic just for 1 second. If you have a question, just have your teacher type it into the chat that you're ready, and then I'll click on you so that we can see you. So maybe while we're waiting for them to let us know, you can talk a little bit about some of the behavior that we saw.
Well, the color changes, of course, are spectacular. Every fish in the spawning mode is black and white. There's no in between. So that's the color change from the normal stripe pattern, the daytime pattern, to the black and white for the spawning is so dramatic. I have never seen this in any other fish.
Okay, so we're going to go to Martha. Will you say that? Sure. We're going to go to Renaissance. Okay, so
They've got on mute. Okay, Martha just asked, she texted her question, what do the grouper eat when they are babies? Well, we can go ahead and ask that question, and maybe you just go ahead and stay on mute. And if you want to do a follow-up, you can either unmute or type in another question. There you go. The answer to the question is, it depends on what kind of baby you're talking about. When those grouper out there at that spawning site, they're releasing their eggs. The eggs are smaller than a pinhead. And within 24 hours, within just one day, they hatch into little fish. And those fish are not much bigger than a pinhead. And for the first 2-3 weeks, those fish are absolutely tiny. And during that phase of their life, they're just floating around and they're drifting with the currents and wind and waves out mostly in the open ocean. And there they're feeding on either phytoplankton when they're very small, or more likely zooplankton, little tiny animals that exist in the plankton. And as they get bigger and bigger, the larger and larger versions of those tiny animals in the pelagic, in the open ocean. And after about 30 days, they've grown to about that big, just maybe bigger than my thumbnail. And that's about the time they start to look for a reef to settle out on. And they use chemical cues. They can smell, and they can also hear. They have well-developed ear bones called otoliths, and those things allow them to hear waves crashing and know to navigate to it. And then they settle out again like the size of our thumbnail. And once they settle out, they start to grow very rapidly, sometimes up to 10% of their body weight a day. After they've settled out, they're just right out here, typically in the seagrass beds and near mangroves, and they're feeding on small crustaceans. As they get bigger, they're about that big. They start to feed on they're voracious. They'll feed on anything.
And they're quick.
Very quick. Yeah. We've one of the reasons why we knew there was a lot of juveniles showing up out here in 2012, as Guy said, was because the local fishermen started catching Nassau grouper this big with flies when they were fishing for bonefish. So they'll eat whatever is rocking around in front of their face, and they need to because they're growing so quickly. But typically it's things like small fish or crustaceans. Yeah, that's a great question. Okay, and out of all the babies, how many actually survive? That's a great question. A given female may have upwards of 1 million, maybe even upwards of 10 million eggs. And in a typical fish population, and that's per year, right? And in a typical fish population, the population itself is doubling if each female ends up producing at least 2 other females that become reproductive over the course of their lifetime. So out of those millions and millions of eggs that a given female produces over her lifetime, you might expect that only 1 or maybe 2 will actually turn into an adult. So the huge amount of eggs that are produced never make it at all.
And it's interesting, Bryce, to contrast that kind of reproductive strategy with, say, sharks and rays that invest all that reproductive energy into a few very well-formed miniatures of themselves when they come out, so to speak. Bony fishes typically will broadcast millions of eggs, hoping that 1 or 2 survive. Sharks and rays will produce 5 or 6 individuals that are ready to go, fully formed, and hope they all survive.
Yeah, and that's what's one of the interesting contrasts for conservation efforts aimed at sharks and rays versus bony fish like Nassau grouper. Sharks and rays, they're very easy to when you start fishing on them, to overfish them, and when they get overfished, it takes them a very long time to recover. Whereas with a species like Nassau grouper, if you set up good protections, that population can recover very quickly. Right. And like they have here, right, over the last 5 to 10 years, the population here on Little Cayman has more than doubled. You know, we've gone from a couple thousand fish to 4, maybe 5,000 fish.
In about 10 or 12 years, which may seem to everybody as a long time, but in fact, it's actually a pretty fast rate of recovery.
Very, very rapid. Yeah. So, Mr. Rogers' classroom asks, "Are there any Goliath groupers out there in these waters?" That's a great question. For those of you who don't know, Goliath grouper they look a little bit like Nassau grouper. They look like a classic grouper, but Goliath grouper can grow up to 6 feet and 500 to 600 pounds in size. They can get very big, size of a small car in some instances. We see them out here from time to time. Yeah, we haven't seen them this year, but in years past, there have been a couple of years where we see Goliath grouper out at the Nassau grouper site. And I was talking to a scientist friend who actually, for a living, studies Goliath groupers. And I'm like, hey, you guys won't believe this. We found these Goliath grouper and they're out there spawning with Nassau grouper. And the researcher was like, no, they're not. It's the wrong time of year for them. They're probably out there looking for a meal. So they're so big that they're the ones that can actually eat Nassau grouper. They were just like the sharks that show up there to eat the Nassau grouper that years. All right, a follow-up question. was, are there more females that survive or males? That's a good question. We don't, you know, this is true of a lot of the things that we do here. We're doing science because we're trying to answer questions that we don't already have the answers to. One of them is we don't have a really good handle on differences in males and females in terms of things like growth and survival for the species. So, you know, it's a good question, and it's actually a really interesting question for fisheries management, but it's one that at present we don't know. We don't know if females tend to live longer than males, and we don't know whether or not on average females to get that much bigger than males. We do know that Nassau grouper, unlike a lot of species of sea basses, they stay the same sex throughout their lives. So they start their lives as male and they end their lives as male, and they never change. There are other species of grouper, like Goliath grouper, that will start their lives as female, but at some point will change to male if they get large enough.
But it's advantageous to have big fat females because they carry more eggs, and the older, the more fecund they become, that is, the more eggs they have. And as you pointed out, the eggs are of better quality too.
That's right. Yeah, and that's exactly why that kind of a question is a really important one, is because, as Guy points out, the bigger females produce many more eggs than the younger females. Yeah.
And talking of luck and survival, I did see Lucky today on the dive.
Oh, yeah.
He was there.
Yeah.
It's quite a big Nassau grouper with a large shark bite out of its back.
Oh,
And actually,
Lucky.
he was swimming over by George. And I got him on film today, so he survived another year.
Yeah.
You could share that with us.
Yeah.
I could post it on the blog because they've seen pictures of Lucky before.
Yeah. So you guys have seen, many of you have seen Lucky before. So it's as Guy said, a fish that many years ago he was a huge chunk of his back was taken out in a big crescent shape. It looked just like the mouth of a shark.
Yeah.
Grabbed its back, shook it, ripped off half its back. But it's a grouper, and they're pretty good at surviving, and Lucky survived. That's why he's called Lucky, because he was lucky to make it out alive. So,
Okay, well, we have a couple of other things to do.
Yep.
Can you say goodbye to everyone on there?
Thank you very much for listening, and hope you learned a lot today. Keep up the great work.
okay, I saw a couple of really great questions, so I want to address them very quickly.
Go for it.
One group of students asked if we've tried transporting grouper from Little Cayman to other places that don't have grouper. We talk about this every year, and it's not something we've tried yet, but that's a great question, because honestly, the scientists are asking the same question, and the managers. So we've been thinking about it. It's going to be a challenge, and there's going to be some logistics to it, but it's something that we have thought about as a management strategy. And then quickly, the last question that I see up here is, how are grouper affected by climate change? And we are actually doing some work with that. The reason why this is important for Nassau grouper is because they spawn when they do at the winter months, because the winter months here, January and February, are the coldest months of the year. So the water temperature is as cold as it gets at any point during the year. And for whatever reason, that appears to be very important for the growth and survival of the eggs that they release during this time of year. And if the water temperatures rise by 2 to 4 degrees, like they're expected to do over the next century based on climate change, then we expect that there's going to be some pretty major impacts to growth and survival of those eggs. We can test that. So we're actually capturing those eggs. You saw, Todd showed that video of the person grabbing the eggs with the bag and showing the camera the bag. And that bag has a unique number on it, so we can then relate the eggs that we bring back to the lab to the fish that we videoed releasing those eggs. And then we can put eggs in different water baths that have different temperatures, and we can simulate the impact of a warming ocean on eggs and see how that warming ocean, that climate change, will affect growth and survival of Nassau grouper into the future.
Awesome. Okay, I see that there's some more questions. We're going to come back to your questions in just a minute, because I have someone else that I want to introduce to you. I also want to say hi to Miss Meisel's class, who just showed up. I can see you guys. Thanks for joining us. When we're done, if you guys want to stay on a little bit longer and chat with me, that's fine. So now I'm going to introduce you guys to another scientist that's working on the project. Her name is Catherine Cameron, and I'm going to let her explain a little bit about what she's doing on the project. And Bryce, you can say goodbye.
Okay, thanks everybody. We'll see you guys soon, okay?
Stay around. All right, so this is Catherine, and she's going to introduce herself and talk a little bit about what she's working on this project.
Hello, my name's Casey. I am a
bit closer to your mouth.
Sorry. PhD student at Scripps Institution of Oceanography. And my advisor and myself study bioacoustics. So we have set up different hydrophones, which are basically underwater microphones, to record the sounds that the groupers are making during spawning. And we've set up multiple of these hydrophones, which will allow us to actually localize where the sounds are coming from and then track the fish as they aggregate.
What kind of sounds are they making, and how did we know that they were making them in the first place?
Well, many of the divers here can actually hear their sounds when diving, and they've done studies other places recording the sounds. And there's different calls that they make. Some are alarm calls. So if they're disturbed or scared, they'll make a certain sound. But during spawning, they'll make sounds that are specific to that spawning. And it's believed that in some way it is either coordinating the spawning activity or might be an indication of who is a good mate.
Okay. And so the end goal of having the hydrophones out there, you were telling me earlier, had something to do with tracking the movement of the aggregation. Have you been able to do that this time, and what is the purpose of that?
Initially, when we first set up our array, we were hoping that we would be able to have the fish spawn within our array and track overnight what they're doing when divers aren't able to be out there observing the spawning aggregation. And this year is actually the first year that we've had spawning occurring really close and in the array. So hopefully when we analyze the data, we'll be able to see what these groupers are actually doing overnight, and whether they're leaving the aggregation or if they have any kind of signs of spawning that are occurring in the night hours when they haven't been observed before.
That's pretty awesome. All right, so could you wave goodbye to everyone? I just wanted you guys to have a glance at some of the other scientists that are working on this project. Thank you, Catherine. And before we take any more questions, I have one more person that I want to introduce to you. Her name is Gracie Simmons, and I taught her in kindergarten, and she has, I know, hi, I can hear you guys there. Hi. And I muted you. I love that. I wish I could do that at school. So Gracie's been working, or she's grown up on this project, and I thought it would be cool if she could share a little bit about what it's like to be a kid and grow up with marine biologist scientists as parents and come on this Grouper Moon project every year and travel around the world with them. So Gracie, maybe you could say a little bit about what your experiences have been.
So I, again, grew up with marine biology parents, so I've gone all around the world, basically just for their work. And so I've got to see lots and lots of different communities and of different places around the world, as in marine and in people.
What are some of the cool places you've been to?
Last year I've been to Cuba. No, not Cuba. Bonaire. Bonaire was last year. Bonaire. That was really cool because it was a lot of other people with reef. And then this last summer I went to the Azores again for the second time. That was really fun. It's a small island near, I don't know, somewhere in the ocean. Samoa, American Samoa. I went to American Samoa and both the other Samoa, just regular Samoa, I think they call it. But that was amazing. We got to stay in these little huts on the beach and just be there, and it's a totally different community. There's like pigs on the side of the road and whatever.
So do you think this has sort of given you a different perspective on the world growing up this way? Because I don't think that most of the kids that are here watching us have grown up the way that you have.
Yeah, definitely. It's been quite an experience growing up with different kinds of people than just in America. America does have a variety, but if you go into other places, there's always more community, as I said, and different like ways of life, which has been really interesting.
And what's it like being around scientists all the time? Like all the time?
I don't know. I don't really know it any other way.
Yeah.
So it's just a whole bunch of chaotic. Oh, where's the giant battery to? I don't know. So.
Okay, well, if any of you guys have a question for Grace, you can go ahead and let me know in the chat, or you can unmute yourself. Or if you have any other questions at all about what we've talked about so far today, now is a great time to do that. I see that Troy Rogers had asked, what laws are there, if any, to protect these fish? And they actually have recently passed a ban on fishing at the spawning aggregation site during the spawning season. So they are protected right now. No one is allowed to be out there fishing them, and that goes across all three islands on the Brack and on Grand. Although there aren't any big aggregations on Grand, there are a couple smaller ones on the Brack. So those they're supposed to be protected right now, so that they can spawn, have more babies, and make sure that we have fish into the future. So that's a great question from Mr. Rogers' class. Do we have anyone? I haven't had any question from Spot Bay. Mr. Stewart, if you guys have any questions, that would be awesome. And I know Miss Meisel's class, you guys joined a little bit late, but you can hop in there if you have any question that you want to ask. Now would be the time. Okay, so say goodbye to Grace. She's going to take off. I'm going to get Bryce back over here to help field any more questions that you guys have. Thank you, Gracie. Maybe done. There we go. Okay.
Hi, everybody.
So, Miss Damon, your class asked, how does the government enforce the protection laws like the ban that I was just talking about? I was explaining how there is now a ban on fishing Nassau during the spawning.
Yeah, that's a great question. Here in the Cayman Islands Department of Environment, which is most of the classrooms here know who that is because you're in the Cayman Islands. But if you were in the United States, for instance, it's kind of like the Fish and Wildlife Service. There is a side of the Department of Environment that does science, and then there's a side of the Department of Environment that does enforcement. And they have officers, and those officers are in charge of enforcing laws and rules that are put in place. And that's how they do it here. They have Cayman Islands Department of Environment, or DOE, officers that have boats that are out on the water and enforcing rules and regulations, whether or not that's rules about lobsters and not being able to take lobsters out of season, or in the case of this time of year, Nassau grouper. And that enforcement is key, though, because if you don't have that, unfortunately, some people will break the rules, and even the best of legislation is not very useful unless you have some ability to enforce it. And the last thing I'll say about that is that it's not just up to the enforcement officers. Some of the best protections in the world when it comes to the marine environment or elsewhere are due to enforcement based on community acceptance and self-policing, or having societal pressure in place where people just don't go because they know it's the right thing to do. So that's equally important.
Awesome. And I see we have a follow-up question. Hi, Mr. Stewart. If you guys have a question, please let us know. Ms. Damon's follow-up question is, what are the consequences for fish, for people who fish grouper out of season?
So if you were to get caught, if you went and fish grouper when you weren't supposed to.
Right.
That's a great question. I believe that if it's a first offense, there's an associated fine. And if you've done it before and you get caught again, the judge who hears a case has discretion to impose higher fines, possibly even jail time, and also confiscation of the boat that you were caught using to take the grouper illegally. So it can be quite steep. It's not something to mess around with.
Do we know if anyone has ever been fined?
There have been some people who have been prosecuted for fishing during the closed season. When there was rules in place, put in place by the Marine Conservation Board, those rules were place-based protections. You weren't allowed to fish Nassau grouper in a particular area. This is the first year that the Cayman Islands government has had legislated rules associated with not being able to take Nassau grouper anywhere in the Cayman Islands during the winter months, during the spawning season. You also are not allowed to possess Nassau grouper nor sell it. And during the rest of the year, even when it's not spawning season, you're not allowed to catch them on spear, and you're not allowed to catch them on pot traps. And then there's a bag limit, or a number of fish that you can catch per day. All of those rules are brand new as of this year, and all of them were put in place based on the science we've done here. We didn't come up with the rules. These were rules that were developed by the Cayman Islands government using the information that we collect. And that's our role here, is to generate information that we serve to the Cayman Islands government so they can make reasonable and prudent decisions about how to manage their own resources.
Awesome. Okay, and I see we have a question from Miss Meisel's class. Hey, you guys, good that you were able to join us. The question is, have the genetics shown that all of the grouper are from Little Cayman rather than Cuba or any other place?
Right. Yeah. No. Great question. The genetics actually shows that there are three loosely different populations of Nassau grouper in the Caribbean basin as a whole. There's kind of a northeast, southeast, and then a Mexican Caribbean population. But it's really, really hard to detect differences genetically speaking. And what that means is actually there's a lot of connectivity among these populations over many generations. And that's not to say that all the fish showing up here are from elsewhere. And in fact, quite the contrary. It's highly likely that the vast majority of juveniles that are showing up on these reefs are coming from the spawning aggregations here locally. It just means that some small portion of fish are coming from elsewhere, and they're interbreeding in the population here locally, and that's causing our populations to look a heck of a lot like populations in Cuba and in the Virgin Islands. Why is that good? Well, that's good because these big aggregations that we've got here, based on genetics, clearly have the potential to seed recovery in populations elsewhere throughout the Caribbean basin. So it's a good news story, not only because we've got healthy populations here, but these healthy populations have the obvious potential to be able to reseed recovery elsewhere in the Caribbean and have the population of Nassau grouper throughout its whole range recover as a result.
Wonderful, wonderful. Okay, well that is technically our time is up for our live video feed. I want to thank Bryce for joining us and all of you guys for joining. I know Miss Meisel's class, you guys just joined now. If you want to stay on, and if my school there wants to stay on, I'll stay on and chat with you guys for a little bit. And with Miss Meisel's class, I can show you some of the video footage that we shot of the Nassau spawning last night. But why don't you guys wave goodbye to Mr. Simmons, who is going to be getting ready for the next dive, I think.
Yeah.
Awesome.
Thank you, guys. All right. We'll see you soon.
All right. So I'm going to stick here for a little bit. Do you, Miss Meisel's class, do you guys want to see the video of the spawning? Do you want me to show you that? Can you give me some? Yeah, thumbs up. Okay. So RSAR, you're just going to have to hold tight for a minute while we watch this video one more time. Okay. See if I can make it work. Hold on 1 second. Okay, it had gone away for a second. I found it now. We will try sharing. And there we go. Okay, so we're going to watch. I'm going to show you guys a couple of different clips, so just watch. Okay, and then this is another cool spot. That is a lot of grouper. All right, so wasn't that awesome? That last shot there where all the fish went right up to the diver was pretty amazing. I think that was shot by Steve Giddings, who is a scientist with NOAA in the US, and he's been working on this project from the very beginning. So if you guys have any more questions, you can ask. And I see Ms. Damon's class is asking, do you have any numbers yet on how many are spawning this year? We're still collecting data, but let me ask a scientist really quick. Christy, is there any estimation of numbers this year?
I don't know the other one. It doesn't stay in my ear. Hi. We don't have the official numbers yet because the scientist who we work with, who analyzes all the count data, doesn't want to give us any information so that we're not biased when we're working on our collecting the data. So we don't know yet, but it certainly seems like there are more fish down there than I personally have ever seen. A lot, as Todd and Bryce were saying earlier, a lot of the young juveniles, or they're not juveniles anymore, the young 5 or 6 year old fish are starting to show up. And so there's a lot of numbers there. That brood stock, like they were talking about, is really going up. And you can't see me. We don't know where it is.
Hold on.
Can you still hear me?
We lost our. Oh, give us 1 sec. I don't know why it's not letting us. say that again because we were muted.
Okay. So as far as the numbers, as I said, we don't, we're the scientist who's analyzing the data as we do the counts don't actually, she doesn't give us any information as we're going along, but we all kind of assume that there's a lot of fish, way more than have been there in the past years. So at the end of the project, middle of next week, Todd will update you on what our numbers are showing in terms of how many are there.
Awesome. Well, you guys remember to, you can go.
Okay.
Thank you so much, Christy, Miss Simmons, Dr. Simmons, Pattengill Simmons. All right, so thanks for being here, you guys. Remember tomorrow, for you kiddos in the Bahamas, we're going to be doing a live dive, and it will start at 1 o'clock. And we're going to go on a dive from a famous dive site here on Little Cayman called Bloody Bay Wall. And I'm hoping you guys can join us. And then my students in Seattle, you can watch if you just go to the blog and click on live streaming. If it's starting at 1 o'clock here, it will start 3 hours earlier there, which would be what, 10 o'clock. Yeah, so if you aren't doing anything tomorrow at 10, you should join us and you can see the live dive. All right, thanks so much, you guys, for being here, and I'll talk to you all soon. Bye-bye. Bye, Miss Meisel's class. Thanks for being here.